Hi, I'm Angelina Jorgenson. I will be presenting on meta-analysis of cardiac extracellular matrix proteins, and this is information extraction for 3D bioprinting. So the issue that we are working to address is based around the modeling of myocardial infarctions. Approximately every 40 seconds, an American experiences a myocardial infarction. These are commonly called heart attacks, and they occur when prolonged absence of blood flow results in death of cells that make up cardiac muscle. This absence of blood flow usually is caused by injury or blood clots. We focused on when it is caused by a blockage in the left coronary artery, which is illustrated in the photo I put on my slide. The major pathology that results is the formation of scarring, and these scars lead to a stiffening of the heart and a composition change of the extracellular matrix proteins. So this stiffness change and extracellular matrix change causes the heart to function suboptimally, so naturally treatments are explored. Animal models are most commonly used for this research. However, they cost a lot of money, and some animals can't even accurately recapitulate the necessary characteristics of human myocardial infarctions due to inherent physiological differences. And also, just from a moral standpoint, any opportunity to avoid animal sacrifices should be maximized. So our lab is working to address this by creating a 3D bioprinted model of a myocardial infarction-affected heart. So as previously stated, the overall goal of the project is to build an in vitro model of MI. We want to engineer a myocardial infarction mimicking tissue for high throughput testing. This is obviously a really big undertaking, so my role in the project was smaller than the overall goal. My first objective was to collect information on scarred and healthy tissue. I specifically focused on extracellular matrix composition and mechanical properties of acute and chronic myocardial infarctions. This information was collected with the purpose of informing the blueprint for our 3D bioprinting. And my second objective will be to compare Bayesian optimization and design of experiment approaches, and I will complete that as I finish my internship into the fall semester, so that will be discussed later in this presentation. So for this blueprint that I was to create for my first objective, I was searching for ECM protein changes with respect to time, which would serve as a template for our design. This was based on information from review papers with a focus on collagen, fibronectin, periostin, and elastin. Collagen is mainly studied as type 1 or 3, but type 4 and 5 showed changes, so I logged those as well. And to demonstrate the trends that I found across all these research papers, I compiled the data into a heat map. A heat map is a mode of data representation where different data values are represented by different colors. Mine is showed on this slide. So for my heat map, for the ECM protein expression, the more red the cell, the higher the percent of the protein present. On the same token, the more blue the cell, the lower the percentage of data. So to present the ECM composition of my heat map, I divided it between acute and chronic. And for both types of myocardial infarction, I reported the relative ratio of major ECM proteins. And for the stiffness, I was reporting fold changes instead of actual values. So before I talk about the trends illustrated, I just want to clarify that for all of the data that I'm presenting right now, I had to go through the process of normalization because all of my sources, they use different means of calculating protein expression. So I needed to convert the data to a percent to create the standard. But now that's prefaced, I'll talk about the trends. So for the protein expression of an acute myocardial infarction, you can generally see an increase in collagen 1 as the red cells of my heat map are getting more red over time. Collagen 3 seems to increase for the first 2 weeks, but then decrease for the remaining 2 weeks. And elastin seems to decrease over the 4 weeks post-MI. Finally, collagen 4 seems to decrease while fibronectin seems to increase. However, those last two do not have as many data points as I would have liked, so it's not as apparent. And for chronic myocardial infarction, finding data was really difficult, but based on what I have, collagen 1 seems to increase after 5 weeks, but decrease after 12 weeks, while collagen 3 decreased after 5 weeks, but increased after 12 weeks. So it's like the opposite. And finally, periostin doesn't seem to change from the base time after 12 weeks. And so as I did for the previous data sets, I had to normalize my stiffness data as well. So for the fold changes, I looked at the stiffness levels of each individual time point and divided it by a baseline or a week 0. And the normalized data, which is presented in my heat map, reveals the expected trend of the ECM stiffening. And you can see that through the heat map. By reading down from week 8, you can see a lessening of the blue color, and that means that the fold changes to the baseline are increasing, which means that the ECM is stiffening. 9 and 10 both only have one color, so seeing that trend is definitely harder, but it displays a stiffening. So in the future, the data collected on the heat map will be used to fulfill my second objective. And as previously stated, I plan on fulfilling this objective as my internship moves into the fall semester. My main goal in the fall is going to be to compare the success of design of experiment approaches with Bayesian optimization. So for design of experiment, I'll start from a model with parameters and find parameters for target viscoelasticity. Then we'll make normal and infarct tissues with the ECM proteins that we identified using 3D bioprinting. And for Bayesian optimization, at each round of experiment, a computer program will calculate uncertainty and next steps to minimize the number of trials, and we will make the identical structures. So the hypothesis in this comparison is that the data-driven approach will reach the optimal point with a fewer number of experiments, and we're going to be able to measure the success by counting the number of trials to reach optimal stiffness and by measuring stiffness using an oscillating rheometer. And so the ranges of ECM proteins given by my heat map will be used for this, which is how my work in the spring semester will connect to my future plans and goals. That's it.
All right, thank you, Angelina. Let's start with Dr. Lima.
Hi, Angelina. That was a great presentation. A lot of information, and the way that you connected your initial objective to a further one. Why is optimal stiffness important?
Because we want optimal stiffness so our heart can function as best as possible, because the result of a myocardial infarction is that the heart will function suboptimally due to an increased stiffness. So we want to reduce that and change it back to how it was previously.
And your research is a key for figuring out how to do that in what way?
Because we're going to figure out, so we're going to look at the two different optimization methods and figure out which one is best for modeling myocardial infarctions. That way, so we can learn how to model optimal stiffness and learn what treatments will reach it best.
Okay, very good. Thank you.
Dr. Constant?
Dr. Lehman got my questions. Now I'm hanging here. The references for your material were shown on one of the slides, right?
Yes, they were.
Just wanted to confirm that. And what's on the oscillating rheometer?
An oscillating rheometer basically measures stiffness. I can't really explain exactly what it does since I haven't used it yet, but basically what it does is it measures stress and produces a stress strain curve based off of that.
That's pretty good for not having used it yet. I don't think I've used one either, but that sounds good to me. Okay, that's all I have. Thank you.
Thank you, Dr. Constant. Boz?
I'm going to ask you the same question I asked last time. What part of this presentation gave you the most trouble putting together, and how did you solve that challenge?
Honestly, for me, I'm gonna say that presenting the heat map was most difficult just because it's a lot of data.
Can you
and
go
I
to
feel
that
like
slide,
it's very
please?
easy to make that. Wait, excuse me, could you repeat that?
Can you go to that slide, please?
Oh yeah. Just the heat map and the trends present, because I feel like it's a lot of data, and that, you know, once you start talking about that, I feel like people just like their eyes tend to glaze over and they just get bored of it. So I tried to present it as quickly as possible, but it was just really difficult for me to like put that all into words, all the trends that I saw.
So you say you were trying to get through it as quickly as possible. So you wanted to get past it was the strategy, or?
Not really get past it, but I just didn't want to go into like too much detail than what was necessary, because I feel like there's no point in like overloading on like the individual squares in each heat map. I just wanted to state the overall trends, that way you guys could get the necessary idea and then move to my central point rather than dwell on like the little details.
Okay, thank you.
Thank you, Boz. We have time for one more quick question. If any of the judges have one more that they didn't get to ask.
What was the most important thing you learned about yourself from doing this project?
Honestly, probably that I'm a lot more patient than I expected. Since when it comes to schoolwork, I have a tendency to take shortcuts and go the easy way. But for research, you can't really take many shortcuts. You have to actually look through every single paper to find all the necessary data. And I realized I'm a lot more patient because I did all of that, and that took a lot of time.